Okay, Dr. Stephen Vogel is on set. He is the senior investigator at the Laboratory of Biophotonics and Quantum Biology at NIH's National Institute of Alcohol Abuse and Alcoholism. His laboratory develops new fiber optic and microscope-based assays using FRET, fluorescence polarization, fluorescence correlation spectroscopy to study protein-protein interactions in living cells. I'm going to keep this short, Stephen, I promise. I'm just saying that today everybody is in for a treat. I got to see a preview of his talk, and the only downside was I didn't get to actually hear his talk, just see his slides. So everybody now gets to hear about his talk on using time-correlated single photon counting of fluorescent proteins to explore quantum behavior in chromoproteins under physiological conditions. Stephen, if you share your screen.
Let's see. Okay, can you see that?
Looks fantastic.
Okay,
Thank
great. Well, first I'd like to thank you, Adila, and Holly, and Feather, and Wolfgang, and Jens for running this great workshop and for foolishly inviting me yet another year to participate. And again, Holly, your morning remarks, if I understood them correctly, you compared us to comfort food. So I'm hoping that I'm an avocado toast instead of a chili mac. So with that said, this image on this opening slide, you know, I like it, you know, and I show it. And I think it encapsulates something that virtually all of us have experienced, and that, you know, we study things in biology, and we try to understand what we're seeing. And most of the time, hopefully, we can figure out what we're seeing and do more experiments and understand the system better and better. But every now and then we see something that's really, really strange, and it's a real head scratcher, and we're trying to understand it, but we can't. And recently, you know, quantum biology has made an emergence, and I'm going to talk a little bit about it, but there's a negative side that I've seen to quantum biology, and that's that there's a lot of people who make this equation. They say, what I'm studying is complicated and I don't understand it and it's not intuitive. And quantum mechanics, I don't understand and is not intuitive. So perhaps the quantum mechanics explains why I don't understand my system. And that's a far reach. So nonetheless, with all of that said and done, I'm going to tell you a story today about some experiments we did that was a head scratcher and the path that it took us down. So my lab, you know, has studied and used FRET quite a bit. And there's two kind of flavors of FRET, heterofret and homofret. And Perry gave a good introduction, mostly of heterofret. And they're essentially the same type of reaction, you know, where you're exciting one fluorophore, and if another fluorophore is nearby, that energy can hop from the first one to the second one, and the second one gives off the photon. And this has been a useful tool in biology called the molecular ruler. So, you know, we published the paper about 12 years ago, but hopefully you can see my cursor here. You know, so typical FRET theory deals with one donor, here blue, and one acceptor, here in yellow. And we did this study, we started it probably 15 years ago, on how will FRET with fluorescent proteins behave if instead of having one donor and one acceptor, what will happen if we have two acceptors? So that was the experiment. And this is the front of the paper, and this is one of the sets of experiments. But we made these four constructs, so let me run you through it a little bit. So all of these are trimers of fluorescent proteins. The blue is cerulean, a blue fluorescent protein. The yellow is Venus, a yellow fluorescent protein. And these whitish translucent fluorescent proteins is a thing we created in our lab. It's called amber, and it's a point mutation in Venus that knocks out its chromophore. So an underlying idea in FRET is that when you have FRET, all of the molecules, all of fluorophores are acting independent of each other, right? So what does that mean? So again, the idea of this experiment, what we were asking is, I mean, you can think of this kinetically. So in this molecule, you excite the blue and the blue gives off a photon. So there's a rate of emission. That's the emission rate. And in this case, where we have one donor and one acceptor, in addition to that emission, you also have energy transfer from the blue to the yellow. And you can think of it as a rate of transfer from this blue to this yellow. But then we made this molecule, and you can think of this one having a rate of this blue to this yellow. So the question is, how will this behave, right? Now, Fret theory says if all of these are acting independently, then this molecule should, the rate of Fret should be this rate plus that rate. That's what it means to be independent. So we did the experiments, and I'm not going to run you through the numbers, but the bottom line of what we found here was if this is the rate to the first Venus, the blue is the rate to the second Venus, then this is the sum of the two. But when we measured it for the ensemble, it was bigger. And it was almost a third bigger. It wasn't a little bit bigger, right? So what's going on, right? So this has been our head scratcher. And again, we published this 12 years ago, right? So, so it's been on the back of our minds, and we've made slow progress in trying to understand it. But even 12 years ago, one of the ideas that we explored was this little quote from our summary, our conclusion summary section, where essentially we're saying, well, maybe they are not acting independently. And if they're not acting independently, then we might be able to explain this extra stuff. So I'm going to go a little bit more into what that means. So now I'm going to say, why should you care? I mean, why care, right? So this is why, right? So I've been listening to talks yesterday and today, and the vast majority of the talks I've listened to have used some version of fluorescence. Not all of them, but many of them. And not surprising, it's a microscopy meeting, right? And when we do an experiment as a biologist, we have certain assumptions that are usually quite reasonable assumptions. So for example, when we use a fluorescent protein, but this applies to any fluorophore, endogenous or exogenous, when we use a fluorophore as biological tag in an experiment, our assumptions about how they behave, are they correct? We have some ideas of how they're going to behave. And if those ideas are wrong, then our interpretations might be wrong. So I'll give you just a couple of examples. So for example, if one fluorescent protein emits one photon at a time, will two emit two? Seems reasonable, but maybe not in a quantum world. Or is the absorption spectra of two fluorescent proteins the same as the spectra of one? Well, that's certainly an assumption of most of us make, but that might not always be true in a quantum world. Or when one fluorescent protein bleaches, does that influence a nearby fluorescent protein to also bleach? Or does the blinking of one fluorescent protein influence the blinking of another one? Our underlying assumptions of all of these things are everything acts independently and they're not influencing each other. And hopefully I'm going to convince you that that's not always true. Okay, so classical versus quantum physics. So this is a nice little book. It's a general audience book on quantum biology. I recommend it. It's a quick read. And for anyone who's interested in trying to understand the gist of this, it's a good, fun, quick read. With that said, the underlying question that every quantum biologist asks is, is biology, when, if ever, do you need to invoke quantum physics to explain something? And there's a lot of physicists who say you never need to do it, right? And this is a little razor, maybe we can call it Steve's razor, which is saying if non-trivial quantum behaviors are observed in a biological system and they cannot be explained by classical physics, then we are compelled to explore if and how quantum physics can help explain the biological behavior. So there's a lot of ifs and ands and thens. And there's also this thing that is prevalent in quantum biology called non-trivial quantum behaviors, which is really a cop-out because who's to say what's trivial and what's not trivial? So even that is not that satisfying. With that said and done, here's the outline of the talk. I'm going to talk about strange things that happen when fluorophores get close. I'm going to talk about venous monomers and dimers and our anisotropy studies. Then I'm going to talk a little bit about quantum superposition and circular dichroism spectroscopy. And then I'm going to talk about photon anti-bunching of venous monomers versus dimers. Okay. Good. So this is a picture of young Foster, who Foster resonance energy transfer, for those who took Holly's survey, working out actually the quantum theory of Foster resonance energy transfer. I don't have to explain it too much because Perry did. But essentially, what Foster's saying is strange things can happen when fluorophores get close to each other. And this was a diagram that I've altered a little bit from a 1964 paper of his, where he outlines 3 regimes of coupling between fluorophores. This is Coulombic coupling. And the weakest, which he called the very weak coupling, is this little box that I've drawn here, which is the fret regime. And then there's an intermediate regime called the weak coupling. And then there's a stronger coupling regime. And the main thing to point out here is that only in this very weak coupling regime are the fluorophores acting independently. Once you go over this barrier, you're now going into a coherent energy transfer regime where one molecule actually influences another one, and actually it starts to become hard to distinguish them from each other. Okay, so how does concentration alter venous A206 dimerization? So GFP and its derivatives, of which venous is, that whole family, there's a particular mutation at position 206, which is normally an alanine in wild-type GFP and wild-type venous. And when you have this alanine, these 2 fluorescent proteins will dimerize, and they exist as a dimeric construct. Roger Chen and his lab years ago played many games with mutating this because they felt it was more useful in biological applications to work with monomers. And they found that if you change this alanine to a lysine, now that positive charge prevents them from sticking to each other, and you have monomeric versions. But anyway, in the dimer, which is the more native kind of form, if you look at the crystal structure, there's a cartoon of the crystal structure, the distance between the 2 chromophores is 2.5 nanometers. So 2.5 nanometers sounds really short to a biologist, but to a physicist, it's so far apart that you'd say there's no way you could have strong coupling that would give you these strange behaviors we're hypothesizing. Okay, nonetheless, we took the monomer, the K206 version, and we made dilutions of them, and we looked at anisotropy. So again, I don't have time to explain anisotropy, but the main point is encoded in these curves are information about molecular rotation, as well as this homofret, this energy transfer. And what you can see is at these 4 lowest concentrations, the curves overlaid perfectly. There was no change. And our interpretation of that is that, well, that's the signal of a monomer in isolation. But then when you got to higher concentrations, now you started to get some curvature and more curvature. And so all of that started happening above 110 micromolar. And our interpretation of that is this is homofret. And that's based on this idea of a relationship between concentration and distance. There's actually 2 curves here because there's some controversy of which is the right one. The green one is more conservative. It's the one my lab has more faith in. But at 110, you're at around 10 nanometers distance, or the 100 angstroms that's famous in fret. Okay, so presumably that curvature is the fret. When we do the same exact experiment with the A206 version, the one that can dimerize, we see again that at the 3 lowest concentrations, the curves superimpose, and that's the monomer. But then as we go above that 1.2 micromolar limit, which is much lower than what happens here, then we start seeing the whole curve shifts down. And then eventually we get to the point where we get curvature. right? So this shifting down, our interpretation is the thing is in fact dimerizing the way Roger Chen's lab said it was dimerizing, and that dimerization starts happening above 1.2 micromole. Okay, so this sets up our next experiment. And our next experiment is looking at circular dichroism. So again, normally with one fluorophore, you have a transition from the ground to excited state, and it's these transitions that determine the absorption spectrum. So if you have two of them and they're weakly coupled, you'll have no change in the spectrum. But if they're not weakly coupled, you have this phenomena called Davidoff splitting, where essentially you're having a quantum superposition of the two excited states. And so now it's splitting those signals, and that splitting of the signals is essentially altering your absorption spectrum. And it turns out that the best way that you can see this is looking at CD, circular dichroism spectroscopy. This shows experiments that we did there. So the black curve is actually 3 curves. That's the monomeric version. Regardless of the concentration we used, it gave the same spectrum. The dimeric version, however, below 1 micromolar had the same spectrum as the black. But now when you went to higher concentrations, when we've already shown they start dimerizing, now you see the spectrum is changing. And if you want to see the Davidoff splitting, you look at the different spectra between the blacks and the reds, and you see this classical couplet. And the observation of this Davidoff splitting couplet indicates that you have a quantum superposition between the two excited states. So from this experiment, we know that we don't have very weak coupling. We minimally are in the intermediate regime of coupling that Foster speculated on. So then we went to the next question, which is to say, okay, well, now that we know that we're pushing the boundary into this next regime, does that alter anything? And specifically, when we have a Venus A2O6 dimer, does it act as one quantum entity or two? So once you have this coherence, is it even fair to think of them as two, or is a dimer actually just a new quantum entity? And if so, how do you measure this? So I have to tell you a little bit about photon anti-bunching, which is a technique I'm sure most of you don't know about. But imagine we have 10 fluorophores, and each one can give off 1 photon at a time, right? So now we collect those photons, we shoot them to a 50-50 beam splitter, we have 2 detectors. Well, if we have 10 photons hitting this thing, we expect, half of them to go to detector A and half to go to B. So if we put a coincidence detector, we should see lots of coincidences because some will hit A, some will hit B. Now what happens, however, if we have only a single fluorophore in our observation volume that could only give off 1 photon at a time? Well, under those conditions, this photon is going to hit the 50-50 beam splitter, and it's going to have to make a choice. Go to detector A, go to detector B. So note, quantum mechanically, right now, we're looking not at the wave aspect of light, we're looking at the particle aspect of light. And under this experimental conditions, it behaves as a particle, and that particle has to go to A or B. So if we put a coincidence detector between A and B, we're going to see no coincidences. Of course, we have to then, change this a little bit for biological experiments. So we're using a pulsed laser. This is the idea if we have like a static, one and only one fluorophore on some mica chip or something like that. And this is the signal you expect. This is coincidences on the y-axis versus the delta time between when these 2 detectors detected the photon. And you can see that at a very short delta time, you have 0. That's the anti-bunching, right? Because it can go here or there. On the other hand, if you do it with a molecule that has 2 fluorophores that are independent of each other, now that central peak goes up. Now, of course, in the real world, we don't do things statically. We do things, change screen. We do things in solution. So when we say 1, we're saying on average 1, so it's a Poisson equation. So these curves change a little bit, but the same idea. The central curve is very low, and it goes up when you have more and more quantum emitters. Okay, so how do we do this? We actually developed a new method called anti-bunching FCS fingerprinting. So what's the deal? So FCS measures the number of fluorescent molecules in a sample. So if you look at this asymptote here on the y-axis of these FCS curves, that's inversely proportional to the number of molecules fluctuating in the sample. Anti-bunching, as I just described it, it actually measures the number of independent emitters in the same sample, all right? And if you then ratio the two, you have a technique for measuring the number of independent emitters as a function of the number of fluorescent molecules. And this is just the proof of principle, and this is the curves for 3 different monomeric fluorophores, as well as Monte Carlo simulations in theory, and they all agree really, really nicely. So the technique works. So now I could describe to you the big experiment. So we have 2 controls. This control is the Venus monomer, the monomeric version of Venus, and it's going to be our control for a single quantum emitter. We made this molecule, which is 2 monomeric Venuses, ligated to a rigid 20 nanometer long rod, with the idea that because it has a large separation between the two, these are going to act independently. And then our experiment is another dimer. This is 2 of the dimeric Venuses with a very flexible linker that allows them to dimerize the way they naturally would. And the question is, quantum mechanically, is this going to behave like a monomer, or is this quantum mechanically going to behave as a dimer? Okay, so we do the anti-bunching, and I'll point out here, everything is color-coded. So the monomer is black, the dimer is green, and the experiment is red. So from the anti-bunching, remember, you look at the center, which is at very short times, and you can see that the red curve looks like the black curve, not like the green curve. And in fact, we can do that at many concentrations, and the same thing holds up. So our conclusion is that the Venus A206 tandem dimer behaves as a single quantum emitter, not as 2 quantum emitters. Okay. So in conclusion, what I've shown to you in this, in this short little story is that there's 2 behaviors that cannot be explained by classical physics. The first one is from the CD experiments showing a quantum superposition of excited states. And I'll point out at a pretty far distance, 2.5 nanometers. Then the second quantum behavior is seeing sub-Poissonian photon statistics for a dimer when it shouldn't really be that way. So then the next big question, which we haven't solved yet, is how do fluorescent proteins do that, right? Because they're not supposed to. So I'm going to tell you our main hypothesis is that it's the beta barrel around the chromophore that is doing the magic. And we have 3 sub-hypotheses of what we think is happening. So the first one is based on this idea that I first heard of from Ross and Mackenzie, where the idea is that if you physically separate a chromophore from the bath, the solution around it, you can lessen the influence of the bath. And it turns out that the bath is usually the thing that makes seeing quantum behaviors hard to do. That the bath is like the big corruptor that makes it difficult to see quantum behaviors. So the idea is simple. The further away the chromophore is from the bath, the slower dephasing will be, and therefore the easier it'll be to see quantum behaviors. We also are considering 2 other ideas. So the second idea is shown in this little panel of, again, this is the crystal structure cartoon of a Venus dimer. You can see the chromophore in blue. But now I've added these dashed yellow lines between the strands of the beta barrel. So those are hydrogen bonds. And in crystal structure, in protein structure ideas, hydrogen bonds can act like springs. And if you think of that as a spring, then this whole beta barrel is a giant vibration isolation device that is surrounding the chromophore. And we think that by doing that, it might make dephasing slower and therefore allow
one more minute,
behaviors to manifest. And then finally,
1 minute.
2 minutes? 1 minute. Okay, perfect time. So, and then additionally, we hypothesize that these beta barrels also have vibrational modes. And when you're in Foster's intermediate regime of coupling, this is the mode of coupling where you have interactions between vibration modes and electronic modes. And so we hypothesize that beta barrel vibrational modes might either facilitate or inhibit electronic transitions. So with that said, this is a complex problem. So we started with a biological problem, but now we're dealing really with how does light and matter interact in fluorescent proteins. And to do this, we've broken it down into 3 different sub-ideas: chromophore protein matrix interactions, fluorescent protein bath interactions, fluorescent protein interactions. And this is certainly way beyond my own lab's capabilities. So over this COVID hiatus, we've actually set up this lovely fluorescent protein quantum dream team consortium to try to tackle this problem of like-minded people. So with that said, of course, thank you. And these are the people who did the work, as well as the members of the dream team that hopefully is going to take it on to the next level. And I'm happy to answer questions.
Great talk, Stephen. Your idea of a dream team is pretty remarkable group of scientists over there. I see what you've been doing during the time off. Yes,
that's been my COVID break.
So we have a question over here that says, what is endogenous concentration of GFP in jellyfish? What was the reason to choose Venus YFP versus EYFP with the phenol or leucine in position 64? It's quite a long question, but I'll let you answer that part first.
Okay. So, we chose Venus because, this whole project was a spin-off of FRET experiments that we were doing using the Cerulean-Venus pair. So, so that's why we chose Venus. And of course, Venus is not a natural fluorescent protein. It's a derivative of green fluorescent protein. Nonetheless, the same mutations in green fluorescent protein, do the same things in Venus, right? So then comes the question, well, what's its concentration? So in the jellyfish, GFP is not free. It's in actual little organelles under very highly concentrated and is naturally found as a dimer. And if you look at the whole spectrum of fluorescent proteins, related proteins, most of them are either dimers or tetramers. Very rarely do you find any of them. I don't think there's any example where in the wild they're found as
Vitaly, if you can join us in the Q&A afterwards to ask the rest of your question. I'm going to go to Benjamin for the last question in this session, and he's asking, how rigid is the alignment between the 2 units in the dimer, or is the rotation of each unit slow enough to contribute to dephasing?
Yeah, so we actually, I mean Grace here, my grad student, did some molecular dynamics modeling of Venus dimers. And what's remarkable, of course, it's very computationally intensive. So, so like if you get a, what was, I think the whole movie was maybe 80 nanoseconds worth of motion in 100 picosecond steps. And, and what you see is they're incredibly rigidly bound to each other. So even though it's one mutation breaks it, when you watch the molecular dynamics, they're moving in concert. So I think even though the dimer can be jiggling around at room temperature in an aqueous environment, the relationship between the 2 chromophores are much, much more stable.
Okay, there's another question in here, but I'm going to ask everyone to join us in the Q&A because I think we're going to get kicked out otherwise of the live stage. So I will hopefully see you guys. We can have up to 9 people sharing their screen. So if you come to the Q&A, feel free to turn on your camera and we can pretend like we're sitting and eating lunch together and discussing this in person.